Hi, and welcome to my second video on Chapter 8, Mitosis and the Cell Cycle. In this video, I'm going to cover the cell cycle, how a cell prepares for mitosis, the individual phases of mitosis, and then lastly, the regulation of the cell cycle. So the first topic we're going to cover is the cell cycle. And this is really just like a pie chart depiction of all of the major events that happen during a cell's life. So if we put a star here, it's this point on the pie chart that marks the beginning of a cell's life. As it progresses in a clockwise direction. So a large majority of a cell's life is called interphase. It's really about 90% of a cell's life. Now we can break down interphase into three subphases. The first of those three subphases is called G1, which stands for gap 1. And that's just when a cell is starting to still grow to its full size, just starting to build proteins, interacting with its environment. And then after that, the second part of interphase is a very important subphase called S phase. And it's during S phase that the cell takes each and every one of its chromosomes and duplicates them. Now we'll learn about the kind of like the cellular mechanics of DNA replication when we get to chapter 10. But it's really a lot of work to do because there's 6 billion nucleotides of information that has to be copied letter by letter. And all of that occurs during S phase. And the reason that a cell does this is to start to prepare for mitosis so that there's 2 copies of each chromosome to give to each daughter cell. Then, after S phase is the last part of interphase, and this is called G2 or gap 2. And it's during gap 2 that the cell really starts to prepare for mitosis. And we'll talk about some of the things that it does to prepare for mitosis in one of the future sections of this video. But collectively during interphase, the cell is really carrying out its specific role in the body. So it really depends on what type of cell you're looking at. Let's pretend it's like a cell of the intestines or a cell of the pancreas. Then it's primarily doing its job of expressing genes and building proteins of digestive enzymes, That's its primary function in the body. It really depends on which cell you're talking about. It's living its life at that point. But our focus here for the remainder of Chapter 8 is going to be on this last little sliver of a cell's life. And that is when it starts to undergo mitosis, and then when it starts to physically split in two, which is during cytokinesis. So we can take mitosis, which is, as we learned before, the division of a parent cell into 2 genetically identical daughter cells. We can split this process into 4 phases. The first phase of mitosis is called prophase. The second is metaphase. The third is anaphase. And the fourth is telophase. And these are consecutive, so it goes from prophase to metaphase to anaphase to telophase. A continuous process. So here again, you can see a depiction of the cell cycle, and you can also see the specific order of all of the stages and substages. There's no new information here. The only thing I want to say is that most cells don't go past G1 unless they're stimulated to divide, either in terms of the natural biological clock within the cell that's telling it, it's time to divide, or from like an external signal, like from another cell, or a hormone like a growth factor. So most cells will stay in G1 unless otherwise told to finish and complete the rest of the cell cycle. Here we have a nice picture that kind of takes you through a cell's journey through the cell cycle and through mitosis. It's very similar to the diagram that I drew out for you in my previous video at the very beginning. So I'm not going to redraw this or re-talk about it. The focus of this diagram is just to show you what happens to a chromosome as a cell progresses through mitosis. And the main thing to notice here is first the chromosome has to get duplicated during S phase, and then during mitosis that chromosome is accurately pulled apart and one copy of each sister chromatid will be given to each daughter cell. So we actually already learned this, but my next section is the 3 major events that happen during S phase and G2 that prepare a cell for mitosis. And the first one we already covered, and that is that all of the chromosomes are replicated during S phase of interphase. The second, which we haven't talked about, is that as a cell progresses from S phase to G2 to the start of mitosis, all of the chromosomes start to get really tightly wound up and coiled up. So as you can see here on the left, before G2, all of the chromosomes are kind of unwound. It looks like a big bowl of spaghetti, like the DNA is just all over the place, all tangled up. That's because the cell needs to be able to access all of the genes, during G1 and S. But as the cell starts to get ready for mitosis, it takes that chromosome and winds it up into that classic chromosome shape that you've seen me draw out. So that DNA gets wound around these purple histone proteins. And the main reason the cell coils up all of its chromosomes like that is it's easier to move them around and to organize them and distribute them during mitosis. It's like packing all that information into a box. Now, another term to say, that chromosomes are getting tightly wound up, tightly coiled, is to say that the chromosome is becoming condensed. And the last preparatory event during G2 is that the cell takes a structure called the centrosome and makes a copy of it. So up until G2, the cell, the parent cell, only has one of these centrosomes. But during G2, it replicates it, so it has 2. Okay, so the centrosome is duplicated. That's really important because as we learned in chapter 4, centrosomes are regions where microtubule filaments are built from. And those microtubule filaments you can see here in yellow, radiating out from the centrosome. That's important in mitosis because those filaments are going to be the machinery for lining up and pulling apart chromosomes. So they're going to give rise to this huge structure called the mitotic spindle that's going to do that for us. Another term for these microtubule filaments is spindle fiber. So if you ever hear me say spindle fiber, it means the same thing as microtubule filaments. My point here is that the centrosome duplicates itself during G2 in preparation for mitosis. Here's a nice picture of a plant cell undergoing mitosis, and you can take a look at the various phases. I've also taken the time to label major structures throughout the process. Here we have the same idea, except in this case it's an animal cell undergoing mitosis. Chromosomes are stained in blue, the microtubule filaments are stained in green. Now lastly, I want to give you my drawing of mitosis, so the majority of the remainder of this lecture is going to be me taking you through the individual phases and drawing out the phases of mitosis. And for this drawing, we're going to make up a cell. And we're going to say that cell is a somatic cell that is diploid. And we're going to say it's a 2n equals 4 cell. And I'll explain what that means in a moment. Now, each time I finish a phase in my drawing, I'll jump back to the PowerPoint slides to fill in any missing information. So we're going to start that drawing. But before we do so, I just want to remind you again of the overall goal of mitosis. And that overall goal is to pull apart each pair of sister chromatids. So basically to pull apart a duplicated chromosome so that we can give one copy of each sister chromatid to each new cell. So that's the overall goal. So all of the things that we're going to be doing in prophase and metaphase and anaphase deal with that process. So we're actually going to start before mitosis here. We're going to look at a cell as it would appear during G1. So at this point, we haven't even got to S phase. So I'm going to label some things in black. That's the cell membrane. In purple, that's the nucleus and the nuclear envelope. We have our chromosomes, and I've coded them by color, so blue represents the paternal versions and pink represents the maternal versions. And as I said before, I'm kind of making up a simple organism so it's easier to draw. And we're going to say this organism is 2n equals 4, meaning it has two types of chromosomes, one large, one smaller. And it has a total of 4, because remember, for each type of chromosome, a sexually reproducing organism has 2 copies of it, one from the mom, one from the dad. So you can see that inside the nucleus, 4 chromosomes in total. Now it's a lot easier than drawing out a human somatic cell. You'd have to draw 42 more, right? Because human cells have 46 chromosomes. That's why we're simplifying things. Now I want to label one more thing. This structure here, this whole thing, is the centrosome. And each of these little black lines radiating out from it are called microtubule filaments, also known as spindle fibers. So that's it. Now we're ready for the next drawing, which is we're going to pretend at this point that S phase has happened. So since S phase has happened, that means we've duplicated our chromosomes. So I need to change my colors here to make sure it matches up with the chromosomes that I already have drawn. So a little bit lighter. Something like that. So since we're after S phase, we have to show that all the chromosomes have been duplicated. And so since this drawing represents G2, right, post S phase, we have to show that the centrosome has been duplicated. So I'm going to show that. And there's my second centrosome. So now we're finally ready to move into the first of 4 phases of mitosis, and that first phase is called prophase. Now, one of the most important things that happens during prophase is the nucleus is broken down, because we need to get access to the chromosomes because we want to line them up and pull them apart. So those centrosomes have to be able to get there. So we have to break open the nucleus for that to happen. So here are the remnants of the nucleus, which I'm drawing in purple. Just the pieces of it that are left over. And now I'm going to draw the centrosomes. And one thing that starts to happen with these centrosomes is the microtubule spindle fibers start to elongate. So we start to build more of them. And as they elongate, they get long enough where they actually can latch onto a chromosome and grab it. And remember, the part of a chromosome that a microtubule filament latches onto is called the kinetochore. So I'm drawing the kinetochore as a little black dot. So I'm going to finish the description I have on the left. It says formation of the mitotic spindle begins. And that means that we start to elongate these microtubule filaments. Also, these two centrosomes are going to start to move away from one another. They're going to start migrating to opposite poles of the cell. And eventually, when we get to metaphase, you're going to have like one centrosome on the north pole and the other on the opposite south pole. So at this point, they're starting to migrate to opposite poles. Then I also want to say that the microtubule filaments from each centrosome start to attach to the kinetochores of each duplicated chromosome. And then lastly, as we said before, the nuclear envelope is broken down. So prophase is the busiest of the 4 phases. And think of it this way. All the things that we're starting in prophase are going to be finished in metaphase. So metaphase is like the finished product of prophase. But before I jump over to my metaphase picture, I do want to jump back to my slides, like I said I was going to. And here's the picture of prophase. There's no new information here. Here's prophase a little bit later on. And you can see we're starting to latch onto and we're starting to move around those chromosomes. And now we're ready for stage 2, which is metaphase. I already have this one drawn out. Now you can see the centrosomes are both at opposite poles. And what's happened is each duplicated chromosome is attached to both centrosomes. So one sister chromatid is attached to one centrosome, and its adjacent sister chromatid is attached to the other centrosome. And those centrosomes have lined up the chromosomes at like what I call the equator of the cell, also known as the metaphase plate. And the mitotic spindle is in black, and it basically includes both centrosomes and all of the microtubule filaments radiating from them. And its construction is complete at this point. So I'm going to jump over to my slides. And there's no new information. Here's a real picture of chromosomes lined up at the metaphase plate. You can see here the microtubule filaments. Here's another stained image that's pretty cool. The chromosomes are stained in blue. The microtubule filaments are stained in red. And the kinetochores of each sister chromatid are stained in green. And now we're ready for the 3rd phase of mitosis, which is anaphase. And during anaphase, we really complete our overall objective of mitosis. We actually pull apart each pair of sister chromatids. So I'm going to write that out. So again, each pair of sister chromatids is separated from one another, and then they start to get pulled and reeled in back to opposite poles of the cell, and each opposite pole is going to start to give rise to a new daughter cell. You can also see that the cell also starts to stretch out and elongate. Now you might be wondering what causes these chromosomes to get pulled apart and then reeled back. And the answer has to do with a protein that uses ATP. In red here at the bottom, it's called a motor protein, and it basically carries the chromosome and uses ATP to walk that chromosome backwards along that microtubule filament shown here in yellow. And it pulls and drags that chromosome all the way back to the centrosome. And as it walks that chromosome back, the microtubule filament gets degraded and starts to get chopped up. So those filaments start to shorten. So it's kind of like reeling in a fish on a fishing pole, and the line is the microtubule filament. The fish is the purple chromosome. And if you're interested, there's also an enzyme called separase. If you remember when I drew duplicated chromosomes in the previous video, they had like those little dotted lines that were keeping them conjoined. There's an enzyme in there that goes in there like a pair of scissors and cuts all those proteins so that we can pull them apart. So next we're ready for the last phase of mitosis, which is telophase. And telophase is pretty easy because it's the exact opposite of all of the events that occur during prophase. So in prophase we broke down the nuclear envelope. In telophase we're going to build a new nuclear envelope around each set of newly separated chromosomes. Also, the chromosomes start to become uncoiled again, or uncondensed. And the mitotic spindle is broken down. So all three of those events are the opposite of what they were during prophase. And you can see at this point we're starting to look like two new individual cells. You should also notice that each new cell looks exactly like the original parent cell. And it also is genetically identical, meaning it also has 4 chromosomes, just like the original parent cell. And that's the overall goal of mitosis. Now, one other event that starts during telophase is called cytokinesis. And that's when the cell gets physically split into 2 daughter cells. So cytokinesis means splitting of the cell membrane and the cytoplasm in half. And that is facilitated by another type of cytoskeletal filament called actin. And those actin microfilaments split the parent cell in half into 2 new daughter cells. At this point, we're done with mitosis. We're done with cell division. And now we have 2 genetically identical daughter cells. And on the left, I'll define cytokinesis. So here's our slide on telophase. Here's our slide on cytokinesis, and here you can see an actual cell being split in half. It's kind of like if you have a pair of shorts and it has a drawstring and you're pinching that drawstring, except pretend your waist is not your waist and pretend it's a cell which is primarily made of water and oil. So if you pinch a cell with a strong fibrous protein, you can pretty easily split it in half. So it's animal cells that split in half using actin, like we described. Plant cells do it a little bit differently. They can't actually split in two. They actually have to build a new cell wall partition between the new cells to help split it in half. And now we're ready for the last section. I won't spend too much time talking about it because, as you can see, a lot of these topics have asterisks and they're not material that I'm going to test you on, but it's definitely relevant and interesting. But the first thing I want to do is I want to take the time to show you this video here at the bottom. And I can only show you it through my account using my school email. So I actually have to show it to you this way. I can't just give you a link. And it's really, in my opinion, the best video overview of mitosis. So I'm going to show that for a moment.
Comprised of 5 unique stages, mitosis is a form of division where a cell's genetic material is partitioned between 2 daughter cells. First, during prophase in humans, nucleic chromatin condenses into X-shaped chromosomes, composed of sister chromatid pairs attached at centromere junctions. Concurrently, outside the nucleus, 2 L-shaped organelles called centrosomes migrate to opposite cell sides. As they do so, microtubule rods begin to grow from each, either towards the cell's interior or exterior, forming a web-like spindle apparatus. Next, the nuclear envelope dissolves during prometaphase, exposing the chromosomes to the cell's other contents. Protein structures also appear on both sides of the centromeres, one for every chromatid. Once these kinetochores form, extending interior microtubules fasten to them, with each sister chromatid being tethered to a different cell pole. Mitosis then progresses to metaphase, where the spindle apparatus rearranges the chromosomes so that they are similarly oriented in a fixed row along the cell's equator. With anaphase, kinetochore-affixed microtubules shorten, and sister chromatids, now individually referred to as chromosomes, are dragged apart. These and other microtubule dynamics also elongate the cell. Finally, the chromosomes land at opposite cell sides during telophase, whereupon the spindle apparatus disbands. The genetic material loosens, and 2 nuclear envelopes, one around each chromosome set, arise. During telophase, in a distinct process called cytokinesis, the cell is cytoplasmically divided. Thus, the end result of mitosis is a cell pair genetically identical to their precursor.
Alright, so our last topic here on the regulation of the cell cycle. I want you to realize that one reason we know so much about the cell cycle and mitosis is that it directly relates to cancer. So cancer is a genetic disease of the cell cycle. So it's caused by problems that occur with the cell cycle. So we're going to learn more about genetic mutations in Chapter 10. But it's any random, unfortunate, accidental change to your genetic material. So like changing a nucleotide maybe from where it was supposed to be an A to a C, or whatever. And when you accidentally change your genetic material, you can mess with one of the proteins that a gene codes for. And if you're unfortunate to have a random genetic mutation that affects a gene coding for a protein that's job is to regulate the pace of the cell cycle, that can give rise to a tumor and can develop into cancer. So a cancer cell is one of your own somatic cells. It can be of any tissue that has accumulated one or more mutations that have affected one of your key essential proteins that's job is to regulate the pace and the frequency of the cell cycle and cell division. So if we look over here, there are many proteins, this is kind of like cartoon analogies, but there's many proteins that kind of act as like red lights. And they prevent a cell from too rapidly moving on to the next stage of the cell cycle. So many times a cell needs a signal, like I said, like a hormone or a growth factor, to tell the cell to go on past this red light, to turn it into a green light. So for instance, if you have a problem with a protein that's acting as this stop sign, and maybe instead of acting like a stop sign, it's acting like a green light, that's going to make the cell divide too quickly and out of control. Now, if you look at this picture, this is one common way that our cells are naturally and normally told to divide when they're supposed to, and that is from a growth factor coming from outside of the cell, maybe from another cell, telling this cell to move on in the cell cycle and undergo mitosis. So the way that works is like this growth factor, which is a molecule, binds to the cell membrane of that cell, and it's going to dock onto a receptor protein in the cell membrane. And that receptor is going to pass on this signal to divide all the way until that signal, that message, gets to the nucleus. And then the nucleus is going to initiate changes that allow that cell to start preparing for mitosis, like, okay, let's wind up our chromosomes. Let's duplicate our DNA. So my point here with this is, if you have a mutation that affects any of these proteins, whether it be the receptor or the proteins that relay the message of the receptor, or genes down the nucleus that initiate changes that start mitosis, if you have problems in any of those proteins, it could result in out of control cell division and cancer. So I want to give you a real example. One of the more common forms of breast cancer is caused by a mutation in a receptor in breast cells. And there's two different ways that this can be affected. One way is that the receptor of those cells, shown in green, can have a mutation that causes it to be like hyperactive. So maybe it thinks there's a growth factor telling the cell to divide when there's not even a growth factor there. So it's sending like a false signal to rampantly divide. That's one way. Another way is sometimes that gene can get mutated, so it's turned on too often. So you're building too many of these receptors. So it doesn't take much of that growth factor to get this cell to divide really rapidly. In any case, they're caused by mutations. So there's a name in cancer biology for any gene that codes for a protein that's essential for regulating the cell cycle, and these are called proto-oncogenes. So those are genes that code for proteins that regulate the cell cycle. That's it for Chapter 8, the first half. We'll talk more about mutations later on in Unit number 3 when we get to Chapter 10. Oops, I want to say one more thing, actually. A cancer cell doesn't become a malignant tumor until you have enough cells, somatic cells, growing uncontrollably. And once they reach the blood, your blood vessels or lymph nodes, and start to circulate throughout the body, that's when it becomes more dangerous as a malignant tumor.